In this lesson, I will be talking about immunoglobulin E and its structural characteristics and functions. Immunoglobulin E, or other known as IgE, are antibodies produced by the immune system. If you have an allergy, your immune system overreacts to an allergen by producing these antibodies. These antibodies travel to cells that release chemicals causing an allergic reaction. The IgE antibody is one of the 5 different classes of antibodies made by our immune system. While there are specific IgE antibodies for every allergy, I have specifically researched the effect of peanut allergies and will further explain the process in which IgE is used when an allergic reaction occurs. IgE is a monomer and consists of 4 constant regions. IgE is one of the 5 isotypes of human immunoglobulins. They are IgG, IgA, IgM, IgD, and IgE. Each of these immunoglobulins have their own specific role in the human body. All immunoglobulins are composed of 2 light chains and 2 identical heavy chains. The heavy chain differentiates the various immunoglobulin isotypes. The heavy chain in IgE is epsilon. The C epsilon 2 constant domain is unique to IgE because the C epsilon 2 region binds to the allergy receptors when an allergy is happening. Heavy and light chains are held together by a combination of non-covalent interactions and covalent interchain reactions. The hinge region is held together by only disulfide bonds. Now I am going to explain the function of IgE. IgE antibodies are produced by the immune system when someone is allergic to something. An allergic reaction occurs when the immune system overreacts to something, often in the environment, that's harmless to most people. To protect the body from this perceived threat or allergen, the immune system of an allergic person produces antibodies called IgE. IgE antibodies are found mostly in the lungs, skin, and mucous membranes. They cause mast cells, which are a type of cell involved in the body's immune system, to release chemicals, including histamine, into the bloodstream. It's these chemicals that bring on many of the allergy symptoms that affect a person's eyes, nose, throat, lungs, or skin. In particular, I researched the effects of IgE to peanut allergies. The condition of a peanut allergy is associated with several specific proteins categorized according to 4 common food allergy superfamilies. These superfamilies are called cupin, prolamin, profilin, and BET-V1 related proteins. These peanut allergens mediate an immune response via release of IgE antibodies as part of the allergic reaction. In specific, while studying the IgE chemical released when allergic to peanuts, I came across a study. In the United States, peanut allergies is present in 0.6% of the population. It has not been clarified to what extent peanut intake affects IgE antibody formation. In peanut-sensitive individuals, the amount of IgE in one's body cannot be determined until the reaction occurs. However, in a study done by the U.S. National Library of Medicine, they tried to investigate the development of peanut hypersensitivity in children and adolescents with specific IgE antibodies to peanuts. The more IgE in the body, the stronger the reaction was to peanuts. Later in the study, a 6-year-old boy's IgE levels were measured. Then, when he turned 11, they measured his IgE levels again, and they were less than they were when the boy was 6 years old. This showed that IgE antibodies to peanuts can decrease over time. This study overall shows a direct correlation between IgE antibodies and peanut allergic reactions. The function of a protein is directly dependent on its 3-dimensional structure. Proteins spontaneously fold up into 3-dimensional structures that are determined by the sequence of amino acids in the protein polymer. To show this, here's a step-by-step explanation of how the IgE protein structure is used in its function. First, in order to first develop allergies, the body must be exposed to something in the environment that prompts it to initiate an immune response. These substances, called allergens, can be something we have swallowed, inhaled, touched, injected, or something inherited. Second, when an allergic individual is exposed to an allergen, the body makes a specific IgE for that specific allergy, for example, peanuts. Third, When the peanut allergen enters the immune system, the allergen binds to IgE receptors on the surface of mast cells. These IgE receptors are placed on the mast cells in order to create a reaction for the next exposure to peanuts. Next, when the allergic individual is exposed to peanuts again, the IgE previously created is able to bind to that allergen. Then, the IgE receptors that were first created then cause degranulation. Lastly, as degranulation occurs, it causes the mast cells to release a series of chemicals that orchestrate the allergic reaction.